DNA helicase is an enzyme responsible for unwinding and separating the double-stranded DNA during DNA replication and transcription. It works by breaking the hydrogen bonds between complementary nitrogenous bases in the DNA strands, allowing them to separate. DNA is the genetic material that contains instructions for an organism's growth and function. Chromosomes are long, organized structures of DNA, and proteins are histones, and each cell typically contains a specific number of chromosomes. DNA consists of two long strands coiled into a double helix structure first discovered by Watson and Crick in 1953. The strands are made up of repeating units called nucleotides, each consisting of a sugar, a phosphate group, and a nitrogenous base. The two strands of DNA are held together by hydrogen bonds between complementary base pairs. Adenine pairs with thymine and guanine pairs with cytosine. The AT base pair is held by two hydrogen bonds, while the GC pair has three hydrogen bonds, making GC pairs more stable. Without the breaking of these hydrogen bonds, the necessary copying of genetic material would not be possible. After the DNA strands are separated by helicase, DNA polymerase attaches to the single-stranded DNA template, starting at an RNA primer laid down by primase. DNA polymerase adds new nucleotides to the 3 end of the existing strand, ensuring the new strand is synthesized in the 5-3 direction. The nucleotides it adds are complementary to the bases of the template strand. Parental DNA refers to the original double-stranded DNA molecule that serves as the template during DNA replication. Helicase is an enzyme responsible for unwinding the double-stranded DNA ahead of the replication fork. DNA polymerase is the enzyme responsible for synthesizing new DNA strands during replication. The leading strand is synthesized continuously in the 5-3 direction. DNA polymerase can move along the template strand without interruption because it follows the unwinding of the DNA by helicase, synthesizing the new strand in the same direction. The lagging strand is synthesized discontinuously in the opposite direction, 3-5, due to the anti-parallel nature of DNA. DNA polymerase can only add nucleotides in the 5-3 direction, so it synthesizes short fragments called Okazaki fragments on the lagging strand. DNA primase is an enzyme that synthesizes short RNA primers on the single-stranded DNA template. Topoisomerase is an enzyme that alleviates the tension in the DNA molecule caused by the unwinding of the double helix by helicase. After the RNA primers are removed and replaced with DNA, DNA ligase seals the gaps between adjacent Okazaki fragments, forming a continuous strand. DNA ligase catalyzes the formation of a phosphodiester bond between the 3-hydroxyl group of one nucleotide and the 5-phosphate group of the next. This process seals in the sugar phosphate backbone of the newly synthesized DNA, completing the replication process on both the leading and lagging strands. In replication, DNA ligase specifically acts to join Okazaki fragments on the lagging strand and finalize the new DNA strands after synthesis. Each nucleotide contains a phosphate group, which is a molecule made of a phosphorus atom bonded to 4 oxygen atoms. The sugar is deoxyribose, a 5-carbon sugar molecule. The nitrogenous base is a molecule that contains nitrogen and forms the genetic code of the DNA. Covalent bonding links the nucleotides in a long chain, forming the backbone of the DNA strand. These strong covalent bonds provide structural stability and protect the genetic information encoded within the DNA. Covalent bonds in the sugar-phosphate backbone maintain the integrity of the DNA strand, ensuring the bases are held in a fixed position for proper base pairing and stacking.